Imagine flipping a coin for your very existence.
Right. It's a crazy thought.
Heads, you become a human being. Tails, your cellular machinery just quietly shuts down before you're even 5 days old.
Wow.
And for roughly 50% of naturally fertilized human embryos, that coin actually lands on tails.
It really does. It is the ultimate biological bottleneck. I mean, it's a massive evolutionary failure rate that you and I and everyone listening, we all somehow survived just to be here today.
Exactly. And so today, in this deep dive, we are unpacking this incredible mystery. We're going to look at a landmark 2026 paper published in the journal Cell, where researchers essentially mapped the exact cellular mechanisms of this coin toss.
And for decades, that 50% failure rate was treated like a biological black box.
Right, we just didn't know.
We knew embryos were arresting, just stopping the development.
Yeah.
But we couldn't clearly see the physical mechanics of why.
But now we have the blueprint, down to the individual microscopic proteins and structural anchors that dictate whether a human embryo survives those first 5 days.
We do. And the way they did it was by literally keeping a camera rolling. They filmed over 150 live embryos continuously for 5 straight days.
Which is mind-blowing. But what's really fascinating is their choice of subjects. They didn't just look at human embryos, they also looked at cynomolgus monkeys.
Which is a super crucial detail, actually.
Why is that? I mean, don't we usually use mice for this kind of stuff?
We do, but in reproductive research, the standard laboratory mouse is actually a terrible proxy for human early development.
Wait, really? A terrible proxy?
Yeah, mouse embryos are, they're almost too robust.
Oh, I see.
They have a greater than 90% success rate in reaching the blastocyst stage in vitro. I mean, over generations of laboratory breeding and just through their own evolutionary biology, mice simply do not make the early structural mistakes that human embryos do.
So they're too perfect.
Exactly. Cynomolgus monkeys, however, naturally mirror human meiotic error rates. They share our early developmental flaws.
So to study our failures, we need an animal model that actually fails like we do.
You nailed it.
Yeah.
And to make sure they were studying natural developmental hurdles rather than just starting with, bad materials, the researchers actually filtered the human eggs they observed.
Right. They compared in vivo ovulated eggs, which naturally matured inside the body, against rescue in vitro matured eggs.
Which are eggs that failed to mature biologically and had to be basically pushed along in the lab.
And that comparison acts as a baseline control, right?
Right. The lab-matured eggs gave the researchers a clear picture of what poor baseline quality looks like.
Like what happens in those cases?
Well, for instance, in those lower quality eggs, the sperm and egg DNA often completely failed to meet in the middle to fuse. They assemble 2 entirely separate spindles and essentially tear themselves apart immediately. Yeah. But the researchers' real focus was on the healthy, naturally matured eggs. The big question was, why were those perfectly good eggs still failing 50% of the time?
And to see that, you have to get incredibly close.
You do.
But recording a microscopic cluster of cells for 5 days presents a massive physics problem.
Oh, totally. Phototoxicity.
Right. If you shine a traditional microscope laser on a living human embryo for that long, the light itself will literally fry the DNA.
It cooks it. Which brings us to the tech they used, the dual-view inverted light sheet microscope.
Okay, break that down for us.
So instead of blasting the entire embryo with a cone of intense light, a light sheet microscope fires a razor-thin 2-dimensional plane of light.
Like a scanner.
Exactly like a scanner.
Yeah.
It sweeps that ultra-thin sheet rapidly through the embryo, capturing optical slices without illuminating the layers above or below it.
So the total energy hitting the cell at any 1 microsecond is drastically reduced, saving the DNA.
Right. And they paired that hardware with some absolutely brilliant chemical engineering. They bathed the embryos in nanomolar concentrations of 2 specific fluorescent probes.
And a nanomolar concentration is tiny, right?
Oh, it's absurdly small. You have to picture a few drops dispersed in an Olympic-sized swimming pool.
Wow.
They used a probe called 5-Sur CTX, which specifically binds to microtubules and lights them up.
And microtubules are basically the physical structural cables inside the cell.
Right, the rigging.
Mm.
Then they use another probe, SBY555DNA, to illuminate the chromosomes themselves.
So we have glowing structural cables and glowing DNA, film slice by ultra-thin slice for 5 straight days.
Yes. And immediately, this continuous, unbroken footage overturned a massive assumption in reproductive science.
This is the part that blew my mind. If you look at older textbooks or previous studies, they often claim that the very first cell division, going from a single fertilized cell to 2 cells, is where the fatal errors happen.
Yeah, that was the dogma for a long time.
But why did we think that?
Because older technology relied on endpoint staining. Researchers would essentially kill the embryo at a specific hour, stain it, and take a photograph. Or they'd take very brief time-lapse snippets.
So they weren't seeing the whole movie, just snapshots.
Exactly. And when you only look at snapshots, you miss the dynamic repair mechanisms. The new, unbroken 5-day footage proved that for healthy, naturally ovulated eggs, that first division is actually surprisingly stable.
The chromosomes align properly, the cell splits beautifully.
Right, the first division was basically a total red herring.
Okay, let's do a quick mental checkpoint here for you listening to crystallize the foundation we've just built. Let's do a quick 5-minute recap with some key bullets.
That idea.
Bullet 1. The 50% failure rate of human embryos isn't just because of poor egg quality, and it isn't happening at the very first cell division as we previously thought.
Right, the first 24 hours are pretty safe.
Bullet 2. By using continuous light sheet microscopy and fluorescent probes, we can watch the entire 5-day process without frying the DNA.
Which is a huge technical leap.
And bullet 3. Cynomolgus monkeys are the ideal model because they share our exact genetic vulnerabilities, unlike mice.
Spot on. So, if the first division is safe, the real danger zone, the biological chaos, erupts during the transition from 2 cells to 4 cells. The second mitotic division.
Days 1 to 3. We can think of this as the 2-cell traffic jam.
I love that analogy.
And the imagery the researchers captured here is just wild. They found that in 30 to 45% of the individual embryonic cells, the physical architecture of the cell division just completely breaks down.
It falls apart. We need to visualize how a healthy cell divides first, though.
Okay, paint the picture.
It's essentially a perfectly orchestrated microscopic tug-of-war. You have a bipolar spindle. Picture 2 anchors, one on the north pole of the cell, one on the south.
Got it.
They cast out those structural cables we mentioned earlier, the microtubules, the cables attached to the chromosomes clustered in the center.
And when they attack from both sides perfectly, that's called an amphitelic attachment, right?
Exactly. Amphitelic. You have balanced tension. The north team pulls half the DNA, the south team pulls the other half, and you get 2 healthy new cells.
A perfect split.
But during this crucial 2-cell stage, the embryo suddenly starts forming multipolar spindles.
Wait, multipolar?
Yeah, instead of 2 anchors, 3 or 4 anchors spontaneously form around the cell.
Oh, wow. So now your tug-of-war has 3 or 4 different teams, all casting ropes and pulling the DNA in entirely different directions.
Right, it becomes a spider web of tension.
A tug-of-war spider web.
Exactly the right image, and when you have 3 or 4 poles pulling, you lose those perfect amphitelic attachments.
So what happens instead?
Instead, you get monotelic attachments, where a chromosome is only pulled from one side. Or you get centriole attachments, where both sides of a chromosome are dragged violently toward the same pole.
So the DNA literally gets shredded and pulled unevenly across the dividing cell.
It does. It's chaotic.
Now, this microscopic chaos actually has an immediate, real-world clinical application for anyone walking into an IVF clinic today.
This is where it gets really practical.
Right. Currently, embryologists usually wait until day 4 or day 5 to see if an embryo forms a healthy structure called a blastocyst before transferring it to the patient. It's a long, anxious waiting game.
It is.
But this multipolar spindle data allows for a highly predictive day 2 algorithm.
Exactly. If a clinical embryologist observes a multipolar division happening at the 2-cell stage, the data is definitive. There is a 3-fold decrease in the probability of that embryo ever forming a high-grade blastocyst.
A 3-fold decrease. So spotting this mechanical failure on day 2 is actually a faster, highly accurate predictor of embryo arrest. It basically removes days of uncertainty for the patient.
It's a game changer for IVF screening.
But if we zoom back down to the molecular level, what actually happens to that unevenly pulled DNA? The cell doesn't just stop instantly, does it?
No, it actually attempts to finish the division.
Seriously, with the DNA all shredded?
Yeah, but because of that 3-way tug-of-war, pieces of DNA get left behind in the cytoplasm, entirely outside the main newly formed nucleus.
Like stragglers.
Right, we call these lagging chromosomes. And here is where human biology dramatically diverges from the mouse model again.
Okay, how so?
In a mouse embryo, these lagging chromosomes form flimsy little micronuclei that eventually just rupture. The genetic material is passively absorbed or just ignored by the next generation of cells.
So the mouse is basically like, whatever, and keeps going.
Exactly. It's a sloppy process, but the mouse embryo just keeps happily dividing.
But the human embryo does not tolerate the sloppiness.
Not at all.
When we leave DNA behind, the human cell builds an absolute fortress around the mistake. It forms intact micronuclei wrapped in a highly distinct, thick outer shell called a nuclear lamina.
And that fortress acts as an alarm bell.
Because it's so rigid.
Because the human embryo has an incredibly sensitive, built-in cellular surveillance program. When it detects these thick-walled, intact micronuclei, it recognizes that a catastrophic genetic error has occurred.
It knows something is wrong.
Right. And within 3 subsequent divisions, those affected cells trigger a profound response. They either undergo cell cycle arrest, meaning they permanently freeze their machinery, or they initiate apoptosis.
Apoptosis being programmed cell death.
Right.
The embryo intentionally kills off its own compromised cells to stop further development. And this right here is cause number 1 of early embryo failure.
It is a very active, aggressive, quality control mechanism.
Okay, let's synthesize this second crucial segment with another quick recap. Bullet 1. The real danger zone is the 2-cell stage, where the cell spontaneously builds 3 or 4 anchors instead of 2.
The spider web.
Right. Bullet 2. This multipolar tug-of-war creates lagging chromosomes, and unlike mice, human embryos lock those mistakes in thick micronuclei.
The fortress.
And bullet 3. That fortress triggers a strict surveillance program that intentionally halts the embryo's growth, which can be clinically spotted on day 2 as a predictor of failure.
Perfect summary. We've established the mechanics of the crash, the what and the how.
But the deeper question is the why. Why do these multipolar spindles suddenly form out of nowhere at the 2-cell stage when the 1-cell stage was totally fine? What physically creates those extra anchors?
To answer that, researchers had to look at a phenomenon called stochastic centriole overduplication.
Okay, let's break down that phrase. Centrioles are the physical microscopic structures that act as the ankles for the tug-of-war ropes.
Right. At the 1-cell stage, right after fertilization, the biology is beautifully simple.
Yeah.
The sperm delivers exactly 2 centrioles into the egg.
A tightly controlled package.
Exactly. Those 2 sperm-derived centrioles migrate to opposite sides, creating a perfect bipolar division. 2 anchors, 2 teams.
But once that first division finishes, the sperm's delivery job is done, right?
Right.
At the 2-cell stage, the embryo has to take over its own manufacturing. Each of the 2 new cells inherits a pair of centrioles. Before they can divide again, those centrioles must physically detach from each other, duplicate themselves, and migrate to opposite sides.
And this is exactly where the vulnerability lies. This specific window of time is known as the maternal to embryonic transition.
The biological software is switching over.
Right, because human eggs don't use these centriole structures for their own meiotic divisions prior to fertilization. The regulatory proteins, the biological brakes that control centriole duplication, they just aren't fully installed or active yet.
So without the brakes, the centrioles just start duplicating wildly.
Yes, stochastic meaning random overduplication. Instead of 2 anchors, you suddenly get 3 or 4.
But proving this was an entirely different challenge, right? Because centrioles are infinitesimally small.
Oh, they are tiny. They're roughly 250 nanometers wide. They are literally beyond the diffraction limit of standard light microscopes.
Meaning light waves are physically too large to resolve them clearly.
Exactly.
So how did they see them? This is honestly one of my favorite parts of the paper. They used a mind-bending technique called ultrastructure expansion microscopy, or UXM.
It's so clever.
Right. If you can't build a microscope powerful enough to see the tiny structures inside a cell, you do the opposite. You make the cell bigger.
They literally infused the embryo with a specialized polymer gel. The chemicals in the gel bind directly to the structural proteins of the cell. Then, they just add water.
And the polymer gel swells.
Yep. It expands equally in all directions. It physically pulls the cellular structures apart symmetrically.
It's exactly like taking an uninflated balloon, drawing a tiny detailed picture on it with a marker, and then blowing the balloon up.
That's a perfect way to picture it.
The picture expands, but the proportions stay exactly the same. By expanding the physical footprint of the cell itself, they could finally get in there and physically count the extra centrioles.
Proving beyond a doubt that overduplication was happening.
But in rigorous science, observing a phenomenon isn't enough, is it?
No, you need graded evidence. You have to prove causality. You need to show that you can cause the crash and then show that you can prevent the crash.
So if the problem is a lack of biological brakes, what is pressing the gas pedal?
The researchers identified a specific kinase, which is an enzyme called PLK4.
PLK4.
Right. PLK4 acts as the master regulator. It's the gas pedal for centriole biogenesis.
Okay.
So for their first experiment, they artificially overexpressed PLK4 in healthy embryos. By flooding the environment with the gas pedal, they successfully induced the exact multipolar errors they had observed naturally.
Wow. They proved that driving centriole duplication causes the shredding of the DNA.
They did, which naturally leads to the rescue experiment.
Right. If you know PLK4 is the gas pedal causing the overduplication, the obvious next step is finding a way to lift off the gas.
Exactly. They turned to a synthetic drug called centrinone, which is a known, highly specific PLK4 inhibitor. It acts as the missing brakes.
So they applied it to the embryos.
Yes, they applied a transient, very careful treatment of centrinone just during that vulnerable two-cell stage.
And it worked. The centrinone effectively suppressed the supernumerary centrosomes. It stopped the extra anchors from forming, and the cells divided beautifully with perfect bipolar spindles.
They chemically rescued the embryo's structure.
That is just incredible. Okay, time for our third recap to lock this in.
Let's do it.
Bullet 1. The two-cell stage lacks the regulatory brakes for centriole duplication during the software switchover.
The maternal to embryonic transition.
Bullet 2. Extra centrioles mean extra spindle pulls, which causes the DNA tug-of-war.
And bullet 3. PLK4 is the driver of this duplication. Inhibiting it with centrinone fixes the spindle structure completely.
It is a massive breakthrough. But as profound as that is, it only solves half the mystery.
Yeah, unfortunately.
We've established all this early arrest mechanics, but even with perfect bipolar divisions, even with perfectly intact DNA, another massive group of embryos still fail.
Roughly 40% of the embryos that survive the two-cell stage flawlessly, with no lagging chromosomes and no multipolar spindles, they still inexplicably arrest and die on day 4.
Day 4. This is known as the morula stage, right? Right before the embryo is supposed to hollow out and become a blastocyst. If the day 2 early arrest is a blueprint sorting error, like a problem dividing the DNA itself, what is happening on day 4?
To figure this out, the team had to switch from looking at structural cables to looking at the factory floor.
The factory floor?
Yeah, they utilized single embryo proteomics, specifically an advanced method called SOVIP, to identify and measure over 6,500 different proteins inside a single microscopic embryo.
Analyzing 6,500 individual proteins in a ball of cells smaller than a grain of sand is just an astonishing technical feat.
It really is. And what the factory floor revealed was a catastrophic meltdown inside the endoplasmic reticulum.
The ER.
Right. The endoplasmic reticulum is the cell's protein folding factory. It takes the genetic instructions from the nucleus and physically manufactures, folds, and ships the proteins the cell needs to build its structures.
And in the human embryos that arrested on day 4.
The proteomic data showed their ER was completely overwhelmed. They were suffering from massive ER stress.
The factory was literally shutting down.
Exactly. And the specific proteins that stopped rolling off the assembly line explain exactly why the embryo collapses.
The researchers noted severe drops in proteins named ZYX, TJP2, PCDH1, OCLN, and PIR-D6B. Now that sounds like a meaningless alphabet soup to most of us.
It does. But these proteins have very specific vital jobs. They are the structural mortar and the architectural blueprints of the embryo.
Okay, give us an example.
Take TJP2 and OCLN, which stands for occludin. These are tight junction proteins. They literally waterproof the outer layer of cells.
Waterproof them.
Yeah. To become a blastocyst, the embryo has to pump fluid into its center and expand, kind of like a water balloon. If you lack tight junctions, the fluid just leaks out. The embryo can't expand.
That makes perfect sense. What about the others?
Then you have PIR-D6B, which is a polarity protein. It acts like a cellular compass. It tells the cell, hey, this membrane faces the outside world and this membrane faces the inside.
So without the tight junctions to hold the fluid and without the polarity proteins to tell the cells how to orient themselves, the embryo just cannot differentiate.
Right. It remains just a disorganized, leaky clump of cells. It cannot organize into the complex structures needed for implantation in the uterus. It just stops.
But how do we verify that the ER stress is the direct cause of this structural failure and not just a symptom of an embryo that's already dying for some other reason?
They went back to the mouse model. Remember how mouse embryos naturally breeze right past the day 4 morula stage without issue?
Right, they're too robust.
The researchers took perfectly healthy mouse embryos and exposed them to an antibiotic called tunicamycin.
And tunicamycin is a chemical that specifically induces protein unfolding. It artificially forces the mouse's endoplasmic reticulum into a state of extreme stress.
Exactly. And when they applied the tunicamycin, the healthy mouse embryos suffered the exact same molecular fate as the failing human embryos.
No way.
Yes. Their junctional and polarity proteins plummeted, they couldn't waterproof their cellular walls, and they arrested perfectly at the morula stage on day 4.
So it cleanly proves that ER stress is the primary mechanical driver of late embryonic arrest.
It does. It validates the entire mechanism.
Okay, let's do a final synthesis of this late-stage hurdle, our final bullet points.
Huh.
Bullet 1. Day 4, morula arrest happens without any DNA or chromosome errors. It's a completely separate obstacle.
Right, non-genetic.
Bullet 2, it is caused by an overwhelmed endoplasmic reticulum, or ER stress.
The factory shuts down.
And bullet 3, the stressed ER stops producing the crucial waterproofing and compass proteins needed to build the blastocyst.
That's exactly it. And seeing human early development broken down into these two highly distinct, physically observable mechanisms, the day 2 centriole overduplication and the day 4 ER stress, it completely changes our clinical perspective.
It really does. It takes the vague, agonizing diagnosis of poor embryo quality that IVF patients have heard for decades and replaces it with concrete, targetable mechanics.
Exactly. If we know PLK4 drives the spindle chaos, we can potentially utilize PLK4 inhibitors in the clinic.
And if we know ER stress causes the morula arrest, we can research chemical mitigators to help the cellular factories keep producing those vital junction proteins.
We're looking at a future where assisted reproductive technology might dramatically alter that brutal 50% coin toss.
But the very mechanics of these discoveries force us to look at the bigger biological picture. The sheer aggressiveness of the human embryo's response is profound.
It really is.
Right, the embryo isn't just passively dying. Unlike mice, human embryos have evolved a highly sensitive, strictly enforced surveillance program that actively initiates programmed cell death the moment it detects a lagging chromosome wrapped in a micronucleus.
Which is a very heavy concept to grapple with. If this surveillance program is so intentionally and aggressively built into our fundamental biology, we have to consider whether this 50% failure rate represents a biological bug or an evolutionary feature.
And that is the exact thought we want to leave you with today. If human biology intentionally halts development to prevent severe genetic disorders from propagating, what does it mean to intervene? By using drugs like centriole to chemically force that developmental coin to land on heads, are we bypassing a vital natural quality control mechanism? Are we ignoring the deeply evolved reasons why the coin wanted to land on tails in the first place?
It's a huge ethical and biological question.
It is a question that bridges the gap between mechanical cellular biology and the very philosophy of human existence. Think about it and let us know where you land.